Welcome to today's EMSL LEARN webinar. We're so glad that you could join us. And our next EMSL LEARN webinar is on February 21st, and it's going to focus on the 1,000 fungal proteins project. For more information will be provided through our social media channels and email for all of our upcoming events and opportunities. So make sure to keep in contact with us through our channels. Today's webinar is going to focus on our metabolomics technologies to advance your research. And as I mentioned, in February 21st, we will have our next webinar. We'll focus on the 1,000 fungal proteins, and keep in touch with us through our social media channels and our email to learn more. And for today, our speakers are Natalie Munoz, an EMSL chemist who will walk you through the metabolomics capabilities at EMSL. Natalie is going to give a brief overview of metabolomics and then also discuss gas chromatography-mass spec. Carl Weitz is here to talk you through atmospheric monitoring and using real-time mass spec. Priscilla Lawley is going to describe the best ways to harness liquid chromatography mass spectrometry. And Robert Young is going to introduce how to use NMR, or nuclear magnetic resonance, for your metabolomics analysis. There will be time at the end of this webinar for questions, so we encourage you to post your questions in the Zoom chat, and our speakers can answer them during the Q&A. And so with that, I turn it over to you, Natalie.
Hi, Linda. Thank you very much for the introduction. So I'm going to start. Okay. Today I'm going to be providing an introduction to metabolomics and gas chromatography mass spectrometry used in metabolomics. So what is metabolomics? Okay, so metabolomics, I'll just go back here. Metabolomics is a field of life sciences that uses statistical methods and analytical techniques, advanced analytical techniques, to characterize the metabolome. The metabolome is in turn the complete collection of the metabolites, or small molecule chemicals with a molecular mass of less than 1,500 daltons that are found in given organelles, cells, tissues, biofluids, and organisms. So metabolites can be endogenous as a result of the chemical reactions that happen in organisms, like sugars, nucleotides, organic acids, but they can also include xenobiotics that come from the diet or the environment. Examples of these are phytochemicals from the food, and also pollutants and pesticides. Metabolites are the direct signatures of biochemical activities. They are extremely sensitive to external stimuli and also to internal signals. In the figure on the left, we're showing how the genes, the proteins, and the metabolites are connected. So the genes code for proteins that in turn process and transport metabolites. And in general, the metabolites together give a chemical readout of what is happening in the organism or the tissue or the biofluid. However, studying the metabolome as a whole is challenging. The first challenge is that there are many metabolites. So in this example, we're showing for E. coli and Cerevisiae, there are between 4,000 to 16,000 metabolites. And for an individual plant, the number ranges from 30,000 to 80,000. The other challenge from about metabolomics is that the metabolites have a large chemical diversity. They have many different polarities, and they belong to thousands of chemical classes. They also have a broad dynamic range in abundance, so they are present in concentrations that range from picomolar to millimolar. To date, we have to use a combination of analytical techniques in order to cover the metabolome as comprehensively as possible. So in the lab, in EMSL at PNNL, we have mass spectrometry techniques, and we have nuclear magnetic resonance-based metabolomics. Using these different type of equipments, we can do untargeted metabolomics, in which we're trying to measure as many metabolites that are present in your sample, and we can also do targeted metabolomics. in which we focus in specific metabolites that you're interested because of some pathway that you modified, for example. We can provide relative semi-quantification or relative levels for the metabolites present, usually for the untargeted metabolomics, but we can also do absolute quantification when we use standards and give you a specific concentrations for this metabolite of interest. It's important to note that from the same sample, multiple analytical techniques and omics can be done. We will, in this presentation, we will cover GCMS, real-time mass spectrometry, LCMS, and NMR-based metabolomics. And each presenter will show the different pros and cons that these techniques have. So I will start by GCMS, or gas chromatography-mass spectrometry. In GCMS, we have here on the right, you can see the GC side, and the analysis begins with the GC, where the sample is vaporized and the components are separated using a capillary column. So we have the sample in these vials. We inject a very small amount, usually a microliter, and this is heated immediately, and the vapors start moving through this GC column. The compounds are propelled by an inner gas carrier, in this case helium, and this is what we call the mobile phase. The separation occurs in this GC column that looks as you're seeing here. This is a GC column, it's very long, and the compounds elute from the column at a different time based on boiling point and also the interaction to the coating on the column. So these columns in the inside, they're covered by chemicals that interact with the metabolites, and depending on the of how strong this interaction is, they elute faster if they don't interact that much, or they take longer. So this is the first part of the analysis with the GC. And then after this, we move to the vapors or the metabolites that are part of your complex mixture are moved to the mass spectrometer side. And they are, the first step that happens there is the ionization and fragmentation. We do this by using electron ionization. This is the most standardized method. It's very mature, and it has allowed the generation of libraries because it's very reproducible too. The fragments that are generated after this fragment, after this step in the ion source, are accelerated through the instrument mass analyzer. That in our case is a quadruple. We have a single quadruple instrument. And in the final step, the ions that are present in the sample are detected, and we get some signal, and we get signals depending on the ions that are present in the sample. So this is in general what happens and the fundamentals of GCMS. When we go to the workflow, what happens when we get your sample? So we get the sample. It can be, we mentioned before, it can be a cell pellet. We can analyze media. We can analyze leaves, soil. So the samples are extracted using MPLEX, which is a modified Folch extraction. We use water, methanol, chloroform, and we get these two phases. In the upper phase, we have the polar metabolites, and in the lower phase, we have non-polar metabolites. For GCMS, we need to dry the extract. We usually use, depending if we're sharing with another technique, we use all of the polar phase and usually half of the non-polar phase. We dry the extracts in a glass vial, then we proceed with derivatization, that I'll explain in the next step. And immediately after that, the sample are acquired in the instrument. Once the whole batch is collected, we proceed to analysis. So I will dedicate a bit more time to this derivatization and analysis step. So why do we have to do derivatization? So the problem is that many metabolites of interest are hydrophilic. They have high boiling points, and due to this, they're not suitable for GCMS. We have to do chemical derivatization to increase the volatility of the metabolites and to reduce the hydrophilicity. The most common derivatizations used, derivatization is just another name for a reaction, are methylation or trimethylsilylation. And TMS, which is shown here with this red group, is what we use in the lab. This is one of the downsides of GCMS because we have to perform this step, and this makes the sample preparation a little bit longer than for other techniques. And how does the data collected look? So when you're in front of the instrument, you start seeing that a total ion chromatogram is being generated. In this case, it's for an extract of bacteria, of Pseudomonas butida, and we have all of these different peaks that represent the compounds that are present in your sample. We can see a signal intensity as function of retention time. However, we have to remember that they elute depending on the interaction with the internal coating on the column. These are some example of metabolites that we observed. But we have to remember that we also have the MS dimension, and we can see here that we get a characteristic mass spectrum for each time point. So the signal intensity is in reality a sum of all of the fragments generated at a specific time point. This mass spectrum acts as a fingerprint, so we can compare the experimental mass spectrum of each of these peaks to a library or a database, and we can confidently say that this corresponds to citric acid. Right now, the PNNL database includes more than 1,000 metabolites characterized by their retention time and mass spectrum. And the type of data generated in an on-target metabolomics analysis by GCMS, we have this list of metabolites with accurate ID. We also present the unknowns or unidentified features with the standardized name, the chemical classification, and the peak areas. It is with these peak areas that we can proceed with statistical analysis like principal component analysis or heat map analysis, and we can figure out which metabolites are contributing to the difference between the strains or the conditions that you were using in your experiment. Finally, some practical considerations for GCMS. So depending on the sample type, we require different amounts of sample. We need around 50 milligrams of wet weight for cell pellets, in order of grams for soils, and at least from microliters to mil for liquid samples like media. Because of its medium throughput nature, as I was mentioning, the derivatization step takes time. Studies of less than 100 samples are recommended. Another issue is that GCMS is a destructive technique. Therefore, if we're using other platforms, if you're interested in LC and NMR, these have to be mentioned soon in the project, so we know that when we're preparing the sample, we have to share and split the sample with other techniques. And finally, that the current PNNL database includes over 1,200 metabolites, but we also search the data with other libraries like NIST and Wiley. NIST alone has more than 300,000 metabolites collected. The mass spectrum is collected, so we search against this one and we can provide tentative assignments for your data too, based on mass spectrum. So this is the summary for GCMS, and the EMSL GCMS metabolomics team is presented here. And if you have any question, please reach out by email. Thank you. Next, Carl is going to be talking about another mass spectrometry technique.
Thanks, Nathalie. Hello all, I'm Carl Weitz, chemist here at PNNL. We're going to talk a little bit about the user capability housed in the EMSL facility called real-time mass spec, or RTMS. We'll run through a few applications using stable isotopes. I don't seem to be able to move anything. Hello?
You don't have control yet, Carl?
No, I don't have control.
Go ahead and click your mouse and it'll turn on for you. So you have to stop touching.
Ah, we have it. Okay. Okay, so we're going to run through a few applications here using stable isotope, which give you ideas on what's possible with your research. What is it? It's basically GCMS system minus the GC. We attach an inlet manifold system to the inlet. It's a split design manifold that not only allows for optimized flow into the instrument, but it also allows optimization of a process sampling flow rate. The figure in blue here is the Battelle patented technology that enables our real-time mass spec capability. The instrument can be used for many monitoring tasks, such as catalyzed reactions, carbon trapping, solvent efficiency tracking, but its most popular use is observing living systems and how they interact with their environments. These are a few of the most recent applications and the experts here at EMSL that I worked with who assisted me in developing these applications. Should be noted that we can, that this has very fast scan rates, so we have great temporal acquisition. We can, depending on the system, we can scan anywhere from 300 to 600 scans per minute. So we can capture most of the gradual respiration responses from these living systems, which is pretty cool in itself. So, and the, oh, sorry, I guess I popped one ahead here. Oh, okay. Oh. Okay. No, that's not there. I'm missing a slide. Sorry about that. Anyway, this configuration here is the simplest scheme for monitoring living systems. The point here is that we can isolate a living system, plants, soils, bacteria, fungus in a stable environment, and manipulate that environment in real time to get real-time feedback, which is pretty cool. The flow diagram here shows how we used RTMS to unveil the Birch effect. If you're not a soil scientist, you probably don't know what that is. Well, the Birch effect is simply the re-wetting of a dried soil, which triggers a rapid release of osmolytes, or in this case, we see here, oh, here we go. Or in this case, we see here the osmolytes by the graph here on the side. It's believed that the microbes store these osmolytes during drought periods to combat osmotic stress. The phenomenon here, as you see it, was first observed and supported by soil scientist H.F. Reported by soil scientist H.F. Birch in the early 1950s. At the time, his contemporaries thought it was nothing more than a reaction of water with soil carbonates. Birch always held the belief the effect of the biological response. So after 70 years, we used RTMS to reveal the Birch effect and actually what it is. In addition, we can now place a time signature on each of these events. So as you can see, we hydrated at time 0. We actually complicated the Birch effect here, where we actually added glucose to our dosing solution of water, which gave us a response of glycolysis, which is also added into this graph. So you can see at time 0, microbes are absorbing water to regenerate. At 38 seconds, you see the Birch effect pull off here at the number 1 point. At number 2 point, at 47 seconds in, we can detect the water that we delivered, which is kind of phenomenal. You would assume that we would detect water first before the Birch effect, but that wasn't the case here. At 56 seconds, 13 CO2 production was detected from glycolysis. At 0.4, at 67 seconds in, you can see microbes start to uptake oxygen. 0.5 at 73 seconds, microbes that fixate nitrogen begin their work, which is that's pretty cool. And the one thing that what Birch had to put up with 70 years ago, I had to put up with a coworker, and he basically said, "Well, that's neat, but the CO2 produced is just a reaction of water with soil carbonates." So I had to put together a scheme to kind of resolve this once and for all. So what I did a stable isotope experiment here where I used basically repeated the Birch effect experiment, but I hydrated with 18O water. So if the Birch effect was a reaction of carbonates, we would expect the incorporation of the 18 oxygen in the initial CO2 burst. We should see that as mass 46 CO2. What we found is that after hydration, the initial burst of CO2 was all 44 carbon dioxide. So there was no incorporation of the heavy water until normal microbial respiration began seconds later. And that's when we finally see the mass 46 and 48 CO2 start to emerge. Therefore, it's kind of cool that after 70 years, we were able to confirm Birch's belief. And if we look here off to the right-hand side, if we sum all the peaks together, we actually come out with our original Birch effect peak that we discovered back 5-6 years ago. So this is kind of a simple monitoring scheme. So we're going to go forward here to a more complicated monitoring scheme. So this basically has all the same elements of the Birch effect experiment. But here we are looking at the Petri dish that was spiked with fungal spores and monitored for 48 hours. What complicated the scheme is that we added time-lapse video, which we can do that now, and VOC trapping. Our flow rate through the Petri dish was approximately 12 mils a minute, and 0.5 mils went directly into the mass spec, so that's pretty much standard settings. The excess split flow was directed onto a VOC trapping cartridge. After 48 hours, the carbon tube was extracted and analyzed using GCMS to reveal what you see here, this rich background of VOCs that were produced by the fungus. We then can open the mass spectra into each one of these peaks and identify unique fragments that can be further searched in the RTMS data to see if we can determine at what stage in the fungal growth the VOCs started to be produced, which is pretty cool. So now we'll go back to our dry soil experiment, get a little bit creative. So what would happen if we took our dormant soil community and rehydrated it on Mars? So right here in the graph you see here, Marvin the Martian is showing us a comparative experiment where we took the same mixed soil lot and divided it into 2 chambers. Dry soil was acclimated for 7 days in both Earth and the simulated Martian atmosphere, then hydrated with 18O water. We observe and compare in this slide here the first 13 minutes of respiration as these dormant microbes essentially wake up on Mars. Interestingly, we cannot compare the mass 44 carbon dioxide burst of the Birch effect because the Martian atmosphere is approximately 95% CO2. However, we can monitor incorporation of the 18O water into the post-hydration respiration of 1618 and 1818 carbon dioxide and compare the soil microbes' initial CO2 respiration production rates. This experiment here will probably be the closest thing we can come to see how a life form from Earth might exist on another planet. And finally, here are a few of the living system enclosures that we custom make here on site. So no matter what your monitoring need may be, I'm sure we can come up with something. And with that, I'll close and I'll pass it on to Priscilla.
Oh, hi. My name is Priscilla. I'm gonna talk a little bit about liquid chromatography-mass spectrometry, or LCMS. I'm waiting for my slides.
Don't forget to click first
I
and then
did.
you can advance.
Okay, there you go. All right. So the analysis it starts with the liquid chromatography separation, which basically separates compounds and mixtures according to their different interactions with a stationary phase and with a liquid mobile phase, which is a solvent or a mixture of solvent that carries the sample through the column. And the compounds that have less affinity with the stationary phase, they will travel faster through the column and will generate a peak in the chromatogram with a shorter rotation time. While the compounds that have more affinity with the stationary phase, they will take longer to travel to the column and will generate peaks at longer rotation times. So the second step is the mass spectrometry. In the mass spectrometry, it measures ions in the gas phase. So after the sample is introduced, the first step is always the ionization, and then it measures the mass to charge ratio or M/Z of the ions, and then they are detected and generate a mass spectrum. Mass spectrometry is widely used in metabolomics due to being very sensitive, very selective, and fast. So a key aspect of the coupling between liquid chromatography and mass spectrometry is this interface, which is the ion source. And that is because since the mass spectrometry measure ions in the gas phase, the compounds coming out of the column, they need to be transferred to the gas phase and ionized before entering the mass spec. So the three ion sources mainly used in LC-MS are electrospray ionization, or ESI, atmospheric pressure photoionization, APPI, and atmospheric pressure chemical ionization, APCI. ESI is the most popular ion source for LC-MS. It analyzes compounds that are semipolar to very polar and from small to very large compounds. So I'm going to talk a little bit more about ESI in the next slide. And APPI and APCI, they both require the sample to be first vaporized and then the ionization occurs. APPI uses a corona discharge to produce the ionization, and APPI uses a UV light. And they are both good for very non-polar species until mid-polar species. And APPI is especially good for aromatic compounds like PAH. So talking a little bit more about electrospray ionization, in ESI, the ionization occurs in solution via protonation in positive mode or deprotonation in negative mode. So it means that it requires the metabolite to have a basic or acidic polar functional group. And this is why it's not compatible to very non-polar species. And ESI has mainly two challenges. The first one is the ionization efficiency, which is if you have two compounds and one of them is more basic than the other, they can be in the mixture at the same concentration, but the compound that is more basic will have a stronger signal. And the other challenge is the ion suppression. That is because since the ionization occurs in solution, there's a competition for the charges. And if you have one of the compounds that they have a high ionization efficiency, it can get all of the charges and suppress the signal for the other metabolites. But if you do have a good LC separation, the ion suppression can be minimized unless you have like a contaminant in your solvent. So that's why it's important to take care of the contaminants for LC-MS analysis. And so the last thing about ESI is that it's a soft ionization, and it means that it produces very little fragmentation, unlike EI, the ionization that Natalie mentioned for the CMS. And this is both good and bad. The good thing is that you almost always get the mass for the intact ions, which sometimes you lose in GCMS. But at the same time, you lose the information that the fragmentation give you about the structure of the metabolites. So to overcome this lack of structural information, from the ionization process, we can use standard mass spectrometry or MS fragmentation. So in this case here on the top, I'm showing an MS1 experiment, which all of the ions produced in the ion source, they are separated according to the mass to charge ratio and then detected. And you have a full scan spectrum with the mass for all of the intact ions. And then the second part is the MS2 experiment, where from all of the ions produced in the ion source, only ions with a specific mass to charge ratio are selected, and then they are activated, and the fragments produced from this specific ion, they are measured, generating a product ion mass spectrum with information about the structure of the metabolite. And this product ion spectrum can be compared to standard libraries to help on the identification of the metabolites. So there are two main approaches for metabolomics, targeted and non-targeted. For targeted metabolomics, you have a predefined set of metabolites that you're interested in. So you're usually trying to test a hypothesis, and the experiments, they can be optimized for that set of compounds. So you have a much better sensitivity in this case. Untargeted metabolomic, the goal is a more comprehensive analysis of all of the metabolites in your sample. So it's used for discovery of novel compounds, or if you want to study metabolic pathways, or also studying different metabolic profiles due to different circumstances. So untargeted metabolomics, like I mentioned before, the goal is to measure as many metabolites as possible. And the challenge is, like Natalie mentioned in the introduction, is the tremendous diversity of molecules present in biological samples. So it requires multiple LC-MS methods and ionization modes to cover as many metabolites as possible. And the two main technologies that we use are reverse phase and hydrophilic interaction liquid chromatography, or HILIC. For both of them, the samples after the metabolites are extracted, they are dried, resuspended, and then they can be run either by reverse phase or HILIC, or both, and by positive and negative ion modes. So for reverse phase, we use a C18 column, so a non-polar column. And in this case, the polar compounds, they interact very little with the stationary phase. So they travel very fast and come out right at the beginning of the run. And you can see that they're not very well separated in this case. While the less polar metabolites, they are more retained, and they come out gradually and are better separated in the C18 column. So this reverse phase method is better for non-polar to semi-polar compounds, such as phenolic acids, lipids. And so the other method is the HILIC, which has involves a water layer on the surface of the stationary phase that retains the polar compounds. So in this case, the non-polar compounds, they elute faster and are not well separated. And the polar compounds, they have a better separation. So the HILIC method is better for polar species, including sugars, carboxylic acids, amino acids. So although we do see lipids in our metabolomics platform, we have a platform that was developed and optimized for lipids, that it's our lipidomics platform. It also uses the C18 column, but it has a gradient more optimized for lipids. So it gives you better information and sensitivity for lipids. So we do recommend that if you're interested in lipids, that you request not only the metabolomics platform, but also this lipidomics platform. So after the data is acquired, there's a series of steps that we need to do to process the data and transform the raw data into meaningful biochemical information. I'm not going to go into detail, in all of the steps, but the most important is the molecular annotation, which gives metabolite identification based on the mass, the mass spectrum, and the retention times compared to standard databases. Here at PNNL, we have a database containing over 1,500 metabolites. We have information about retention time and mass spectrum, and we also have access to external databases. So just some practical considerations for LC-mass. It's a relatively fast pipeline, so it is compatible with large-scale studies. It covers a wide range of metabolites. Untargeted metabolites have two choices of methods: reverse phase for semipolar and no-polar metabolites, and helix for polar metabolites. And we do have the lipidomics platform as well. Untargeted metabolomics provide qualitative identification and relative quantitation. So if you are interested in absolute quantitation, we recommend a more targeted approach with the use of internal or external standards. So finally, salty samples, they are very problematic in LC-MS because the salt ions, they have a very strong signal that can suppress the signals from your sample. And also they can form salt deposits on the instrument that cause a lot of problems. So if you have a salty samples, it requires some cleanup steps, and you can lose some metabolites during this steps. Okay, so this is our LC-MS metabolomics team and the LC-MS lipidomics team. Feel free to reach out if you have any questions. And thank you for your attention. And I'm going to turn it over to Robert now.
All right. All right. Hello, everyone. Can you hear me? Okay.
Yes, yes.
Okay. Apparently, I don't have control quite yet. Okay. There we go. Finally. All right. Thank you, Priscilla. I'm Robert Young. I'm a chemist and I specialize in NMR. And so we're going to switch from mass to talk about something completely different, but highly complementary. Okay. My arrows are not working. Oh. It's like there's a delay. Sorry about that. Yeah, so nuclear magnetic resonance capitalizes on the fact that certain nuclei have magnetic properties and interact with strong magnetic fields. In the simplest case, these are spin 1/2 nuclei. They'll occupy, in these magnetic fields, they'll occupy one of two quantized energy states, one higher and one lower. Over here, we have 4 spin 1/2 nuclei that are biologically relevant. As you can see, they're not all equally abundant, and they're also not all equally sensitive in the NMR experiments. Those that aren't very abundant are great for use in stable isotope probing studies, just as they are for mass spec as well. And so the difference in their sensitivities is related to the difference in the two energy levels, and that depends on some isotope-specific physical properties, as well as the strength of the applied magnetic field. Listed here, I have the frequencies for each of these nuclei at our highest solution state spectrometers field. And in the experiment, what we do is we pulse radio frequency into the sample, matching that resonance frequency, and that perturbs the system in such a way that we get a measurable response. And that response comes out looking like this. It's called a free induction decay. And we'll mathematically transform that into a spectrum with peaks at ever so slightly different frequencies, which I'll get to in a minute. To be able to compare data acquired at any magnetic field, we'll always reference these to the chemical shift scale. Before moving on, I just want to talk about some of the key features of NMR. It's a non-destructive method. Experiments are highly reproducible. the peaks that we see are quantitative, and it provides atom level structural information in molecules and often requires minimal sample prep. Its main drawback is that it's less sensitive than relative to other methods. And so in metabolomics, we're typically measuring polar metabolites in water for NMR. And what we can give you is what's there in abundance and thus readily bioavailable. And so the workhorse in metabolomics and NMR metabolomics workflows is the 1D proton NMR experiment. And this has to do with the fact that it's the most sensitive nucleus, and therefore experiment times are the shortest. And thankfully, protons are in most metabolites. And so I've surrounded this 1D NMR spectrum with some 2D, some multi-dimensional experiments that we can also tap into, say, if the spectra are too complex and we need to disperse some of that signal, we'll break into a separate workflow and look at that. Also, if we need to identify an unknown metabolite, we have the tools available to do that. But since standard workflows oftentimes only need the 1D proton experiment, I'll just continue talking about that. So NMR nuclei are very sensitive to their local bonding environment. And so small changes in that will lead to shifts in their peak positions. And as you can see from this proton chemical shift scale, the chemical shift alone gives you a clue as to what kind of functional group these signals are emanating from. There's also other peak information. If you look here, you'll notice splittings. And these splittings that you see in peaks, they're predictable and they relate to the number of protons on neighboring atoms. One of the reasons also the peak areas, if we integrate the peak areas, they're going to correspond, they're directly proportional to the number of protons at that molecular site in the entire sample. So all you need is one compound of known concentration, and you can and you can determine the concentration of all the others. The reason I've selected these two sets of examples here just kind of highlights the complementarity between NMR and mass spec. These compounds, as you can see, have the same mass, and in some mass spec methods, it's not readily apparent which one is present. In NMR, that sensitivity to structure allows us to readily distinguish between isomers and also determine the solution concentrations of them. So with this kind of brief introduction to what we can get out of these spectra, I'll move on to workflow and just talk about what these steps look like in the NMR workflow, from sample preparation up to the initial data analysis. So one advantage of NMR is that it's very tolerant of wide-ranging sample conditions. And so we can make measurements when salt concentrations are high or at extremes of pH. And so a lot of times we can take samples like supernatants from microbial growths or water extracted soils or root washes. We can take those and just measure them directly. And that allows us to obtain information on low molecular weight, volatile compounds that are otherwise lost in subsequent preparation steps for other methods. I kind of highlight here with this example showing a pore water, which is where we take moist soil and centrifuge it to squeeze the water out. A small portion was taken and measured directly, and the remainder was sent down through another processing pipeline, which involved a dry down step, and then subsequently redissolved to concentrate. As you can see, key metabolites like ethanol, methanol, acetone, acetate, those are lost upon the dry down. But because NMR can screen the sample with no sample preparation, we can get their identities and their quantitation. And then we can also look at the sample after it's extracted and dried and get a better look at some of the less abundant non-volatile metabolites. So in NMR, we put the samples in a glass tube. And so no matter how nasty the sample is, it's not going to hurt the instrument. And of course, the measurement is not going to hurt the sample. And so if you're really are really sample limited, you can have NMR screen it and then pass that on to other analyses. When it comes to data acquisition, most of the steps in data acquisition are pretty reliably automated, and that includes sample changes, instrument setup, data collection, and at least initial signal processing. Two of our instruments have automated sample changers, and one can even accommodate these racks that contain 96 NMR tubes. And they're also able to be stored at 4C. And so for preservation, they could be stored at cold temperatures before and after measurement. Typical times for a 1D NMR experiment is a few minutes to a few hours. It really depends on the concentrations of those metabolites. Here at EMSL, we have 4 cryoprobe equipped spectrometers with field strengths ranging from 600 to 800 megahertz. Cryoprobes are more sensitive probes than standard probes. Our coverage there includes proton, carbon, nitrogen, all of them to do those. And we also have the capability on some to do phosphorus and fluorine if those happen to be of interest. Just mentioning these isotopes, I want to say that stable isotope resolved metabolomics is possible by incorporating them as tracers. There's going to be an upcoming webinar, I think April 10th, that's going to discuss stable isotope probing in metabolomics, and my colleague David Hoyt will be presenting the NMR portion of that talk. And so moving on to data analysis, there's 2 major approaches to data analysis. The first is, the first that I'm going to cover is the chemometric, also known as global untargeted. In this approach, it basically divides the spectrum into bins or buckets. And there's a lot of nuance, a lot of sophisticated approaches just to how you prepare the data and normalization schemes, things like that. But the gist is divide the spectrum into bins and then take the intensities and see, using multivariate statistics, see if there are any significant differences between samples or groups. Usually this is done with PCA. And then you can look at loadings plots to see where those significant differences are, which bins. And then you go back to the spectrum and search for what metabolites are covered in that in those bins. Overall, this method does not seek to identify or quantify all metabolites, only those that are responsible for significant changes between groups. The most common approach we use here is the quantitative analysis approach, and that is an untargeted approach where we indeed try to identify and quantify as many metabolites as possible. The way we do that is we use feature matching, spectral feature matching, using libraries that can either be commercial, custom, or publicly available, as well as analysis software. The one I'm showing on the screen here is a commercial program called Phenomics. And as you can see here, Trehalose, the peaks from Trehalose are highlighted, and we use this. We can shift these around to see if the spectral features match and adjust the intensities to get the quantitative, to get the concentration. And what we get out of this type of analysis is we provide a list of all the metabolites that we can identify and their solution concentrations for each sample. And then that data would then go and be fed into other statistical analyses or models. In case you're wondering, we can regularly detect metabolites in the low micromolar range. And as I mentioned very early on, we do have tools and separate workflows that can help us if the samples are especially complex, or if we need to go after and identify unknowns. Once we do identify a known, we can add that to our libraries. And so they're constantly expanding. Some practical considerations if you're getting a proposal together. Although NMR is very tolerant of high salt concentrations, if paramagnetic ions are particularly high, that can be a problem. And that might require a cleanup step like SPE. Also, if you're interested in water extractions of soil organic matter, please plan on providing some test samples so we can optimize the water to soil ratios for NMR. And speaking about samples, the typical number of samples you can plan for in a project. I would say if you're in the ballpark range between 30 and 60 per year per project, that's a pretty reasonable range. Always, if you have any questions or want to talk about the technical details of your project, please give any one of us a call. Our contact information is here, or you can navigate to the EMSL website and the NMR Metabolomics page. So that concludes the NMR portion of the talk, and just kind of a step back, just to kind of get a broad overview here. We learned about some significant analytical platforms. They all have their strengths and weaknesses. But as this Venn diagram that contains metabolites identified per method in a very recent root exudate study, you can see that the overlap is limited. So they're all capturing things that no single technique can. And so to really get a comprehensive view of the metabolome, it's best to incorporate all three. And you can do that here at EMSL. And on this slide, we have a quick guide that compares the different platforms side by side, along with their strengths and their weaknesses. It looks like we're a little behind on time, so I'm going to go to the next 2 slides, and maybe during the Q&A section, we can come back to this so you can get a longer look at it. And with that, on behalf of my colleagues and myself, thank you for your interest and attention. And I think we can move on to questions now.
Welcome back, or hello, everyone. So, all right, for the Q&A, before we start that, I just want to quickly remind everyone, we have 2 open call for proposals that are closing tomorrow, or at least the letters of intent are due tomorrow at 5 p.m. And that would be our EMSL's large scale research.
Oh,
Okay. For some reason, I kept being put on mute. So we have 2 open calls that are the letters of intent are due tomorrow. Just FYI, the large scale research and the EMSL ARM FICUS call. So if you are going to submit an LOI, please do so by tomorrow at 5 o'clock. And on to our questions. Looks like we're running out of time. So those whose questions we don't get to, our folks will go ahead or all of the researchers here will go ahead and answer them. If everyone can come back on camera, if you can, that would be great. All right, so Sharon had a question for you, Natalie. Since the PNNL database includes 1,200 metabolites, but microbes have 3,700 to 16,000 metabolites, are efforts continuing to expand your database?
Yes, our database is continually expanding. However, we have to take into account that we cannot, by GCMS, we cannot see all of the metabolites that are present in microbes. Many of these are going to be secondary metabolites or lipids that have structures that are too large, and GCMS had a cutoff of 600 MC. So if we have a higher molecular weight, we cannot see them. And if we cannot derivatize them, we cannot see them either by GCMS. However, if you have metabolites of interest that are not in our library, but you have the standard, we can run it here so we can acquire the retention time and mass spectrum. And then every time that we analyze your samples, we will look for that one specifically.
Great. Okay, Jadesh was asking, Carl, does EMSL have thermal desorption or other VOC analysis platform? And he apologizes if it's an off-topic question.
Well, yes, we have GCMS, volatile capabilities. We have actually a new GC-QTOF instrument with a thermal desorption unit on it. So yes, we do have that capability. It would be under the TAP IRP, and that would be China SWARP would be your contact there. We can pretty much customize any any VOC analysis that you might have. So just let us know what you need and we'll see what we can do.
Okay. Arjun asks, which method would be useful to look at glycogen storage in soils, or is it possible to differentiate glycogen from glucose from mass spec techniques? Who wants to take that one?
I'll take it. It is possible to distinguish between glycogen and glucose, but this is an example where you'd have to use multiple analytical techniques. Our current LCMS Metabolomics pipeline cannot distinguish between certain sugar isomers, whereas GCMS is quite capable of doing so. However, glycogen is a little larger. I believe it's like over 600 in terms of its mass to charge. So you wouldn't be able to see that with a GCMS, where you would be able to see it as LCMS. And as the polysaccharides get larger and more branched, they become water insoluble. So they become insoluble for the most part. So that would be something that would be, you would have to consider. Also with a lot of these polysaccharides, it's hard to distinguish between these isomers using LCMS. So it would be something that we would actually have to, if you're sure that glycogen is the polysaccharide in your largely in your sample, whether and how it exists, then we can accommodate that. But if you're not, then that would require a little bit more hands-on and different approach than what we typically do.
Great. Okay, one last question. And as I mentioned to the audience, if there's any questions we didn't get to, just know we've noted them, and everyone here can, or someone here can answer you back. Okay, so Ravitika, she asks, where do you perform statistical analysis of LCMS data, hydrophilic data set with 13,000 identified metabolites?
Say that again, 13,000 identified metabolites?
Yeah, where do you perform statistical analysis of LCMS data?
So EMSL does have a statistics team that usually assists us. We do use our typical pipeline use is PMART-R that was developed here by really great statisticians. Usually when we do supply users with those tables, sometimes users would request that we do the statistics for them. We definitely can do that if you're not comfortable doing that yourself. But PMART-R, which is I think it's freely available, it's an R package, is the one that is preferred.
Okay. Well, at this point, our webinar is concluded. I hope everyone had learned a lot and had a great time. And again, for those, Sharon, I saw you had another question, and to Desh, and so we will get to your questions via email. All right. Thank you. Have a great rest of your day.